Thank you for that introduction. I'm going to go ahead and share my screen so that y'all can see the presentation. Cody, can you see? Yes, no?
Yes, we can see it.
Okay, great. So as Dave said, today I'm going to be presenting on regional climate and soil health management and how we here in the Southern Plains have been delivering science-based climate services at the Climate Hub. Oh, come on now. So to do a little roadmap, I'll start with just a really brief introduction to the Climate Hubs and then talk about the larger project that I've been doing, which looks at agricultural management practices, climate, and soil health. And then at the very end, I have another couple tinier projects that have to do with farmer use of microbial community data. So the Southern Plains Climate Hub is part of the 10 hub network under the USDA of climate hubs. Basically, if you're in the United States, your state is associated with a hub. The Southern Plains Climate Hub happens to have Kansas, Oklahoma, and Texas. So depending on if you're in SCIP or in the CAST, you may have other states and other climate hubs, so none of them all overlap perfectly. The 10 hubs, it's a cooperation amongst ARS and the Forest Service, and there are also co-directors for each from NRCS, Forest Service, and ARS. And there are 10 fellows. We've been here for all about the same time, about 3 years. The mission statement of the hub is to synthesize and deliver science-based, region-specific information and tools through the existing networks of USDA agencies, which would include like NRCS, ARS, Forest Service, RMA, other partners. And other partners to help land managers make climate-informed decisions. And sometimes that looks like what you see here, which is me out in the field with some folks sampling soil. We do soil health field days, and we often will set up at trade shows or conferences, this is our rainfall simulator right here, with some information about the hubs. Why we exist is U.S. agriculture contributes a pretty good deal to the economy. In the Southern Plains, we contribute, just in the Southern Plains, mind you, we contribute almost $60 billion. So agriculture overall contributes about 5.5% to the U.S. economy. Of that, just farm output is about 1% of the U.S. economy. So it's pretty important, and there needed to be some way to, help land managers make those climate-informed decisions. The main project I've been working on while I've been here has been to do with agricultural management and climate effects on soil health in the Southern Great Plains. What you see here is a picture of the Southern Plains region, Kansas, Oklahoma, and Texas, and that's the land cover, different land cover classes essentially of how this area looks. On the left is I think that's grain sorghum, but don't quote me on it. And then there's, a farm over on the right. If you look more closely at that map, you'll see the crops are this kind of beautiful goldenrod color, which unfortunately is really hard to see in amongst all of the grassland and everything else. So I colored it ugly purple. And you can see that the croplands, they're especially prevalent in Kansas, but they're actually found all over the whole region. And they contribute approximately $16.2 billion to American agriculture. So they're pretty important. Also in the Southern Plains, we have this really neat gradient, both of temperature and precipitation. So you can see that in these pictures. Basically, as you go from north to south, you go from mean annual temperature of about 10 to a mean annual temperature of 25. As you go from east to west with precipitation, you go from about 1,500 millimeters over in the far east all the way to just 200 millimeters over there in the Texas panhandle area. And those two combined give you kind of a northeast to southwest gradient of evapotranspiration, which also affects what farmers can grow and how they can grow it. We know that the climate in the Southern Plains, as everywhere else, is changing. The Southern Plains is projected to get warmer. So under the lower emission scenario, which essentially now is no longer possible, it would have been between 4 and 5 degrees for pretty much the entire Southern Plains. Under the higher emission scenario, you can't really see that two of those reds are combining, but it's anywhere from about 6 degrees temperature change to 9 degrees temperature change. And with those hotter temperatures comes a projected loss of soil moisture, which is important for farmers, especially if they are rain-fed agriculture instead of irrigated agriculture. So a loss of anywhere from about 5% to 10%, some areas looking more even at 10 to 15% under the end of century changes. And farmers have been having to deal with this already, farmers and ranchers. So one of the ways that they improve their resilience now, and they'll continue to improve it in the future, is through some of these new technologies. So that can be things like grass cast, which you see on the right here. That basically was built to tell ranchers the likelihood that they will have more grass or less grass compared to usual for the time period that the map is intended for. So this one was made in July 31st, 2018. And then there are other things like searchlights, which in this case, this is the cattle heat stress alert that they have. And that basically will text ranchers when temperatures are going to get high enough that their cattle might be experiencing heat stress so that they can actually do something about it. Farmers also use new precision planting and fertilizing techniques, as well as precision watering, where instead of just spraying water everywhere, they'll actually water closer to the ground, or some of the newer systems will even water individual plants. I don't know if those are in wide use yet. I imagine not. Another great tool that they have, especially in farming, is changing up their farming practices. So primarily, this includes switching to no-till or conservation tillage. And what you see up in that top pie chart is the conventional tillage compared to no-till or conservation tillage in the Southern Plains. They can also incorporate livestock so that they have a more diverse ways to earn money. If the crop doesn't do as well, they can sell cattle. Also using cover crops, and they can sometimes graze those, or they just improve the soil. And also using diverse cash crops. So instead of growing just one thing, grow lots of different things. Some of this they've been doing for a long time, not just in response to climate, but more in response to how the climate behaves, especially in this region. It does wild things. What I'm mostly focused on for my research and for this talk are these new and modified farming practices. And if you're familiar with the NRCS, you've probably heard of the pillars of soil health. So these are basically to have healthy soil. And their definition of soil health is the continued capacity of soil to function as a vital living ecosystem that sustains plants, animals, and humans. So there are 4 pillars to basically improve and maintain soil health. The first pillar is to disturb the soil as little as possible, which means not tilling it or tilling it very little. The second is keep living roots in the soil as much as possible. So this is where cover crops come in. You want to keep the living roots in the soil because it feeds the microbial communities. Keep the soil covered with vegetation or vegetation residue. Cover crops also help with that. They're planted when the field would ordinarily lie fallow, so they're called cover crops because they keep the soil covered. And then the final one is grow diverse cash and cover crops to encourage a diverse soil biota. And that has to do with the microbial communities, which I'll talk about a little little later. When you do all four of these things, you end up with a system that more closely resembles this is basically a wild prairie that you're seeing with, roots of different diameter and length, and you have this really nice layer of organic matter rich soil right at the top that has a lot of nutrients and holds on to water really well. Past studies have shown that managing for soil health, so using no-till and using cover crops and incorporating livestock, it has been shown to improve soil organic matter concentration, soil structure, water holding capacity, and also the fertilizer efficiency. And a lot of that is mediated through this soil organic matter concentration. So when you disturb the soil less, you allow the soil to actually build up soil organic matter, and that's basically just when it's broken plants down into the component parts, they become soil organic matter. What you can see over in this graph on the right is just a measure of soil stability. And if you look at the solid black bar, that's no-till, and then the hashed bar is conventional till. And you can see that from 0 to 5 inches, which would be the top 2, or 0 to 5 centimeters, which is the top 2 inches of soil, that it's a lot more stable under no-till than conventional till, and the same holds true for 5 to 20 centimeters. And that's from a paper in 1998. So we've known about a lot of this for even further back than that. My objectives with this study, first was to determine the effect of soil health management versus conventional management on soil health of farms in the Southern Great Plains. You'll see SHM and CM show up again, that it's basically soil health management versus conventional management. And then second, to determine the role of regional climate conditions in influencing the effects of soil health management practices on soil health. So basically, does using soil health management always accomplish the same thing in the same amount of time, regardless of climate? You would assume the answer would be no. So just to remind you of this gradient, I was able to make use of it. You can see the sites on there, and I'll have a better map of them in a second, but basically I was able to take advantage of the east to west precipitation gradient and the north to south temperature gradient. I did restrict my sites to only those areas that were using rain-fed agriculture, so that's why they don't go far outside of there, is because once you get outside of that area, you start getting into irrigated agriculture a lot more. So I originally started with these 12 locations. I went out and sampled all of them. One of them didn't work out because it actually turned out after the fact that it was irrigated, so we threw that one out. At each location, I had 3 pairs of fields, so 6 fields total. 1/2 of each pair was soil health managed, and the other field in each pair was conventionally managed. And those paired fields were usually right across the fence from each other. If not, they were within a mile of each other. At each of those 6 fields, I had 3 sampling locations. And at each sampling location, I took both physical, chemical, soil samples, and also samples for the microbial community. Those physical or chemical samples, I took 2 at each sampling location, and those were the big wide cores. They're about 5 centimeters in diameter. The microbial community samples, I took 8 that were more spread out, and those were they're like really narrow, 2.5 centimeter samples. The physical and chemical samples I sent to OSU to be analyzed by their soil, water, and forage analytical lab, SWAFL. And then the microbial community samples I analyzed here at the Grazing Lands Research Lab. For that, I used phospholipid fatty acid analyses, and if that sounds like a mouthful, that's because it is. You'll pretty much the rest of the talk, I will call them PLFAs, because phospholipid fatty acid takes too long to say. They're basically these little things that are in every single pro or eukaryotic cell, so whether it's a bacterial cell or the cells in your body, and they basically keep the inside of the cell inside and the outside of the cell outside, they have these chains that look like legs in that picture. Those are the fatty acid part. It turns out there are a lot of different versions, and some of those are reliably specific to a given functional group or a clade, so a bunch of related, in this case, microbes. So we use them to indicate different types of soil microbes, but they're not unique indicators. So some of them can belong to multiple of the microbial groups or to plants and animals additionally. But they're still really helpful for determining broad community structures. So we're not getting to specific microbes. Like, we're not going to sit there and say, oh, there's E. coli in this soil. But we're able to say, oh, there are this many prokaryotes, there are this many gram-positive bacteria. So it's really just general types of microbes. We did a ton of statistical analyses. So for the chemical physical samples, we did t-tests to compare soil health managed to conventional management. We did Pearson correlations, and we used a p-value of 0.1 instead of 0.05 because there's so much variation. So we did those correlations for environmental variables, which would be temperature, precipitation, and evapotranspiration. And then also percent sand, silt, and clay, which would be soil texture. We also did it for conventional management and soil health management, and then the difference between soil health managed and conventional managed fields. For the microbial communities, we had to treat those a little differently, so we did a principal component analysis to compare soil health managed to conventionally managed and see if they broke out in any specific way. We also did regression using the environmental variables and individual microbial functional groups and split them into soil health managed and conventionally managed to see if those microbial functional groups behaved differently related to the environmental variables. Spoiler alert, our results were that there were no significant differences overall between soil health managed and conventionally managed fields with regards to those physical, chemical, or biological indicators. And that was basically the first result that we got, because that's a very broad level result. I wanted to throw the computer out the window, but I did not. When you start, when you start looking at it more closely, you actually start seeing some really interesting trends. So for example, this is a graph of phosphorus. If you look over along this left side, you'll see in S, that's because these are arranged north to south, and then east over here to west over here. So you start seeing things like, for phosphorus, some of the soil health managed fields, the phosphorus was a lot higher in that upper sample. Some of them, they were not. So this side over here is 0 to 5 centimeters, the very surface of the soil. The other thing you notice with phosphorus that's very interesting is as you go from north to south, you get less phosphorus in the sample just overall. And that could be due to the rocks that the soil came from. It could have to do with how much phosphorus people are applying to their fields. It could have to do with precipitation as well, or temperature. You notice some similarly interesting things with potassium, except in this case, you have both the north to south, it reduces, the amount of potassium reduces, and then also going from west to east, the amount of potassium in the soil decreases. This is also really commonly applied in fertilizers, so I think that's why we're getting some of these kind of somewhat odd results, especially in Hutchinson, Kansas. I think that's because the farmers there were applying potassium. Soil organic matter is one of the most interesting ones and one of my favorites because it mediates so much of the response that we see to different management practices. And what you'll notice is that in the where we're seeing the most differences between soil health management and conventional management is in the upper 5 centimeters of soil. And that's kind of just a a fact of how soils work. Basically, the deeper you go, the less soil organic matter you get, and then also the stuff at the surface is more likely to be incorporating like plant residue and everything more quickly. So when you're going to see changes happening as a result of soil health management, you're going to see them first in that upper 5 centimeters of soil, and then you slowly might start seeing them further down. You'll notice that in multiple cases, this soil health management, it was higher in the upper 5 centimeters of soil, and that happened in multiple environments. So not only did it happen in these really wet areas like up in Esben, Kansas, which is northern Kansas, it also happened in Ardmore, Oklahoma, which is in southern Oklahoma, right near the Red River. And it happened in Bonham, Texas, which is south and east. And it happened to an extreme in Bonham, Texas, which I think is a result of that guy grows a ton of cover crops and he grazes a lot of cattle. And that soil has not been disturbed in something like 40 years. So he's been doing no-till a long time. You see less going on in Vera, Texas. That's because it's really hot and dry there, and it just takes soil organic matter forever to form in the first place. When we looked at our Pearson correlations between the soil health indicators there across the top here and the climate, soil texture, and management variables over here on the left, we saw that temperature especially was affected or was correlated with every chemical or physical indicator, and that was a significant correlation. But then we saw that management didn't affect anything significantly along there on the bottom. So that was an interesting result, and I'll talk a little bit more about that in a moment. For the microbial communities, this is a principal components analysis. The thing to know about these principal components analyses is basically you're looking at how similar these are to each other. So the closer two of these are to each other, the more similar they are. The further apart they are, the less similar. So this guy right here is not very similar to this one right here. And you can group them by doing these principal components analysis. It looks like there are two groups in this. So you have kind of this group over here, and then you have this group over here. What's interesting is that has nothing to do with how they're managed. You don't see all the bulbs to one side and all the regular ones to the other side. This actually, as far as we could tell, is more mediated by precipitation. So you've got the more precipitation over here and less precipitation in this group. When we looked at the relationships between those climatic factors and the microbial functional types, we saw some, positive and negative relationships, but overall there weren't a ton of correlations between them. So with regards to our first objective, which was to determine the effect of soil health management versus conventional management on the soil health of farms in this region, we saw that the management effects were significant only for some of the locations and some of the indicators, for example, with organic matter at 5 centimeters in Esben, Kansas. And those effects were not consistent, meaning that we didn't consistently see higher organic matter in soil health management fields at 5 centimeters. Sometimes we saw it, sometimes we didn't. Our second objective was to determine the role of regional climate conditions in influencing the effects of soil health management practices on soil health. And with that, we saw that climate had a significant effect on multiple indicators as well as some microbial functional groups. And I just realized that I highlighted texture as climate in this graph. But what's yellow from basically here up, that's all correlated with climate. And then over here also, anything that's highlighted was a correlation with climate. However, we did not see soil health management having any effect on soil health, which makes it hard to really tease out how climate affects the effects of soil health management, because first you'd have to have some effect of soil health management to see that. So those of you who have done much reading in this know that this is really inconsistent with previous studies. Previous studies always pretty much find that soil health management does have an effect on soil health. But it turns out actually that the one other study I could find that had a similar, whoop, there go our lights, that had a similar study design actually found the same results as me. So they had a pretty wide area that they covered with their experiment. And they found that climatic drivers were more influential than tillage or cropping in determining the soil organic matter levels. And that's basically an effect of drowning out of the effect of climate versus management. So our sample size, the local sample size, was only 3 pairs, which when you have like a system like this where there's a lot of variation, 3 pairs is really not enough to start teasing out any actual effects of anything. Whereas our regional sample size was 33 pairs, which is a lot easier to start teasing out where you're actually seeing effects. So in summary for this part, the differences in soil health at a regional scale are driven primarily by climate, not by management. And there are some methodological and statistical challenges of studying these field scale differences at a regional scale level. So for example, we would have needed a lot more samples at the field scale, and that would have required a lot more people and a lot more man hours, and not a ton of people have access to that much, time and people. It's important to consider the constraints imposed on soil health by climate, and I think that's the main takeaway, is that you can't just assume that we commonly say there will be at least a 3-year lag between implementing soil health practices and being able to see changes in soil health. Well, that might be true for some areas of the United States, but that might be an absurdly small amount of time for that to happen in like a dry area like Vera, Texas, which it was on the southwestern side of my samples. And that's important because when you tell a farmer that they're going to see something in 3 years and they don't, if using that new practice is harder than using the old one, like if they've had to rent equipment or something, they're just going to go back to the way they were doing it before because it's cheaper, it's what they know. And so we don't want to give them an unrealistic idea of what to expect. And I think that's the most important takeaway that I got from this project. I also wanted to talk about producer use of microbial community data. So while I was out taking samples, I went and I visited a ton of farms and talked to a ton of farmers. If you've gotten to work much with farmers, you know that they love to talk and tell stories. And so in amongst all of this storytelling, we would sometimes talk about the results I was going to get, including the microbial community stuff. And it turns out that a lot of them are really interested in it, the ones I work with. But this guy from Garden City, Kansas, put it really well, and I wish I could remember his exact words, but I've paraphrased him. So he basically said, it's really interesting, but it's also kind of useless. It's really expensive, and it takes too much time for what you get out of it. You get all kinds of information, but what's it actually tell you? You can't do anything with it. So essentially, they get all this information about microbial communities, but there's no way to tie that to how they can actually make decisions about management. So with knowing soil organic matter, we know how they can improve soil organic matter, and we know what low soil organic matter means for a farm. We don't know that kind of thing for soil microbial communities. So while I was analyzing all of my samples, I had a lot of time to sit and think some more. And I thought, well, I can't begin to approach how do we make the data that they get back actually useful? Like, how do we find a way for them to relate it to yield or to soil health or to soil organic matter? But I can at least start looking at this kind of time and money investment thing. So I came back and I thought, well, I'll do something to see what is the actual optimum number of samples for PLFA analysis of a field. how do you get the most information, the most like accurate information with basically the least amount of work. And then actually it turned out that I had a collaborator at KSU, Dr. Gretchen Sassenrath, who was also interested in how we actually make microbial community structure relevant to producers. And so we actually teamed up, and we're going to see if we can relate crop yield to different microbial community structure aspects. And we don't know if we'll be able to. It's kind of in the pilot study phases. So for that first one, for optimizing sample size, these are the directions given on Ward Labs' website. So Ward Labs is currently, I think, the only lab that can do consumer available PLFA analyses. They have these directions, and the one that really stood out to me is this third one, which says take 10 to 15 cores per, essentially per field. Well, I've been to a number of different farms, and those farmers have widely different sizes of fields, and so I was like, well, 10 to 15 samples for what field size? For example, if you have 10 to 15 samples on a 75-acre field, basically you're saying each sample represents 5 acres a sample. On the other hand, if you're in only a 30-acre field, you're saying each of those samples is representing roughly 2 acres a sample. And that's, not perfect given how things actually work, but basically you want to know how many samples you need an acre or something like that is what I really wanted to get to. So the question was basically, is 10 to 15 samples enough for every field, or do you need more samples on bigger fields? Since I already had a bunch of friends out there, I went back to several of the field sites that I used before, including Parsons, Kansas, Loyal, Oklahoma, and Ardmore, Oklahoma. And I collected this time 30 samples each from 3 fields. Each of those 3 fields was being managed with soil health management, so no-till. In Parsons, they don't use cover crops, but in Loyal and Ardmore, they do both use cover crops. And all those fields were kind of different sizes along a gradient from 15 acres to like at least 100 acres. I analyzed each of those samples individually so Ward's directions also say to just combine all of the samples into one. I wanted to combine them mathematically instead of physically. So I analyzed each individually, and I got kind of the results that I would be showing you here. So I can find what percent of the total microbial biomass is in, say, gram-positive bacteria or gram-negative bacteria. And these are the results in a format similar to what Ward uses. So I wanted to use, insofar as I could, Ward's methods, and then also look at their results in as much the same way as I could. I then used statistical analyses and some fun computer modeling to quantify the number of samples necessary to reliably characterize the soil microbial communities in these differently sized fields. And what you would expect, so the way I ran those computer models is I would basically say, okay, I have 30 samples in this field. I'm going to look at, if I ran every possible combination of 2 samples, what would be the variation in like the averages among those? So I would have some really big numbers and I would have some really small numbers. And I did that for every sample size from like 2 to 30. You couldn't do it with 1 because you can't get an average with 1. And what you expect is as you incorporate more and more and more samples, that the variation starts going down. And it turns out that when I did that, that is exactly what happened. So these are preliminary results. I am still working on this one, and I still have multiple fields to do. So these are all from Loyal, Oklahoma, which is just north of here. It's in central-ish Oklahoma. This is for a 40-acre field out there, and you can see that at 15 samples, there's actually not that much variation. So each of these lines represents a run of samples. You can see that, you know, at 15, there's fairly little variation. I might say, yeah, 15 samples would be pretty okay for a 40-acre field. If you look, however, once you go to the 90-acre field, this line is at 10 and I don't know why. So if you look at this 15 samples right here, you see actually quite a bit of variation between all of the samples. To get to the same amount of variation that you had in the one right before it, you'd have to go to about 25 samples, between 20 and 25. And you don't see that big of a change with 100 acres because, you know, the distance between 90 and 100 is just not as big. But again, at 15 samples, you see quite a lot of variation. As coming from a science perspective, I would prefer not to have that much variation. I would much prefer to have the amount of variation you'd get with like 25 samples. And that's just for one functional group. I need to look at it for the other functional groups as well. But that's an idea of what it looks like right now when we've got preliminary results. Basically, right now, it looks like the appropriate number of samples for a reliable representation of the microbial community appears to increase as the size of the field also increases. So basically, yes, you do need more than 10 to 15 samples once you get to a big field. The other kind of half of this project was also undertaken in Parsons, Kansas. That's where my collaborator is. She's with Extension for KSU in Parsons. And we both wanted to know if there's a correlation between microbial community structure and crop yield. That would be the easiest thing for farmers to, you know, have it relate to something is crop yield. The reason we would suspect that this could actually be the case is we know that some microbial functional groups, so for example, arbuscular mycorrhizal fungi, which they help the plant access nutrients, and they are associated with increased productivity of plants, increased drought tolerance, and increased soil moisture and plant moisture availability. So they give more structure to the soil because they're kind of like webby, and so they can hold the soil together better, and that helps the soil hold on to water, and so that's available to plants during drought. We also know that other fungi, as well as some bacteria and other, you know, soil microbes, confer increased protection from pathogens. So basically, kind of like the microbes in your tummy keep you from getting sick, microbes in the soil can keep certain pathogens from spreading. And reduced herbivory, I actually don't remember the mechanism of that, but they've looked at it. And then, you know, there are so many other relationships that could be out there that we just don't know about because these microbial communities the study of them has only been fairly recently in scientific time. So we're still discovering new things about them every day. And now that we have the ability to look at their DNA instead of just microbial functional groups, we're finding out even more. A good example of some of these relationships is this kind of schematic right here. This isn't an actual graph of things. This is a conceptual diagram. So it basically shows that when soil nutrient availability is really low over here, the microbial community diversity is high and its contribution to plant productivity is really high. So that would be especially important in areas with kind of cruddy soil that, you know, you don't have as many nutrients. Conversely, when soil nutrient availability is really high, maybe it doesn't matter as much because microbial diversity is low and its contribution to actual plant productivity is also a lot lower. So it, you know, might not matter quite as much. There's currently little or no information on correlations between community structure and crop yields. And so that's basically the objective, is to determine if we can correlate yield with microbial community attributes. This is still just a pilot study. We're just seeing if we can do anything. And if we find something interesting, then we may try to turn it into a full study, or she may hand it off to a grad student to do, because it would be a really interesting grad student project. But basically what we're doing is we would spread the 30 samples around this field, and then we can look at these yield maps that Gretchen gets whenever they go and harvest. And the red is low yield areas, the green is high yield areas. So basically we're going to look at if the microbial communities in low yield areas have anything in common, and if those are different from high yield areas. So that's going to be exciting, and I imagine it'll be more complex once we actually get into it. And I don't have a summary conclusion slide, but I wanted to thank all of you for being here and listening to me talk. And I would love to take any of the questions that you have.
Let me turn off the screen share as well.
So yes, I'm going to turn off the screen share as well.
Well, thank you very much, Cait. You know, this is